Today we're going to look at the central nervous system. The central nervous system consists of the brain and spinal cord. They're the primary integrators for the central nervous system, or for the nervous system in general, and is comparable to a central switchboard of the telephone system. It receives incoming sensory signals, decides what to do with that information, and sends out the appropriate motor outputs. So we can really think of it as a central switchboard. The brain and spinal cord of the central nervous system are located in the dorsal body cavity. And over the course of animal evolution, cephalization has occurred. Cephalization is simply the elaboration or growth of the anterior portion of the central nervous system. And this goes hand in hand with the increase in the number of neurons in the head that we've seen over a period of time. This increase in the number of neurons in the head has very much occurred at the highest level in the human brain. So we've seen the highest level of cephalization in the human. If we're talking about embryonic development, we must start with the fact that the brain and spinal cord don't begin as the structures you're familiar with. We're not talking about kind of a wrinkly walnut sitting on a stalk. Instead, initially we'll have to talk about the neural tube. The neural tube is pretty indistinguishable. We know it has a front and back, so it has an anterior rostral side, and it's that front portion where we start to see three primary brain vesicles or three brain or neural tube swellings occur. These primary brain vesicles are the forebrain, midbrain, and hindbrain, also known as the prosencephalon, mesencephalon, and rhombencephalon. So here we can see the neural tube, really simple. It has a front and a back. Then we start to see kind of brain swellings, or the primary brain vesicles develop, the prosencephalon, mesencephalon, and rhombencephalon. The mesencephalon will remain unchanged and will eventually become the midbrain. The prosencephalon breaks down into secondary brain vesicles, telencephalon and diencephalon, and the rhombencephalon will become the mesencephalon and myelencephalon. So those structures then will develop further and become the adult brain structures, and within these adult brain structures we'll also have lateral, third, and a fourth ventricle. These ventricles are connected via aqueducts and canals. The central canal is located in the middle of the spinal cord, and the cerebral aqueduct connects the third and fourth ventricle. There are two lateral ventricles located in each cerebral hemisphere, and we'll talk about them in more detail, but they all contain cerebrospinal fluid. Because the brain is growing more rapidly than the skull that contains it, it begins to fold or convolute in on itself. Again, this membranous brain inside the skull is not able to fit with the rate of growth of the skull, and so it's going to fold it on itself and create sulci and gyri. That's why the cerebral hemispheres have kind of a wrinkly appearance. The cerebral hemispheres, the left and right ones, they grow posteriorly and laterally, or they grow back and then to the sides. And during the growth of the brain, so during the development of the primary and secondary brain vesicles and trying to get to the four adult brain structures, we're going to see that during that growth period, two bends occur, a midbrain flexure and a cervical flexure. And this is going to give the brain kind of a more familiar appearance, where we're used to the cerebrum being more superior than the brainstem and the spinal cord. As what we can see here is the midbrain and cervical flexures, which will eventually be able to give the brain kind of the appearance we're used to. And what we're seeing down here at the bottom are the four adult brain structures, which have pretty much developed by birth. And so we'll see that adult pattern of structures and convolutions at birth, and we'll have two cerebral hemispheres, the diencephalon, cerebellum, and brainstem. So at birth, the brain already contains the pattern of four main structures and numerous convolutions that will develop and persist into adulthood. So on any question, test, or assignment, you're asked to list the four adult brain structures. You would list the cerebrum, or cerebral hemispheres, diencephalon, brainstem, and cerebellum. You may also be asked to list the three structures of the diencephalon, and those are the thalamus, hypothalamus, and epithalamus. The epithalamus is also known as the pineal gland. For the brainstem, there's also three structures: the midbrain, pons, and medulla oblongata. The midbrain, pons, and medulla oblongata are being listed from superior to inferior. The medulla oblongata is continuous with the spinal cord. The spinal cord is not an adult brain structure. The diencephalon is medial to the two cerebral hemispheres, and we can see that the midbrain sits right under the diencephalon. The cerebellum is behind the occipital lobe of the cerebrum, and it's sometimes called the little brain. Next, we want to talk about the basic pattern of gray and white matter in the central nervous system. White matter consists largely of myelinated nerve fibers, and white matter is made up of fatty substances. Fat doesn't stain, and histologically it appears lighter than gray matter, which is composed of neuron cell bodies, dendrites, glial cells, things that aren't myelinated, essentially. And the orientation of these two groups of nervous tissue in the brain and spinal cord differ. We actually see that in the spinal cord, there's centralized gray matter. So there's a central cavity surrounded by a gray matter core for the spinal cord. And then external to that are columns of white matter. In the brain and in the cerebellum, it's the opposite. We would see outer gray matter and inner white matter. Especially in the cerebral hemispheres, though, you would have some deep gray matter. And that deep gray matter is called basal nuclei. Here is a better view of the orientation of gray and white matter in the brain and spinal cord. So we can see on the right that the outside of the brain has gray matter, the inside has white matter, and then there's some deep sections of gray matter. Those deep sections are deep gray matter called the basal nuclei. Then we have the deep white matter, but we really want to pay attention to that outer portion of gray matter. That outer gray matter of the cerebral hemispheres is called the cerebral cortices, or sometimes just the cerebral cortex. Then over on the left, we have the spinal cord, we have the outer white columns or funiculi, and then we have the gray matter horns. Ventral and dorsal gray matter horns are common terminology when talking about the gray matter of the spinal cord. Our next topic is the ventricles of the brain. The ventricles are hollow chambers that contain cerebrospinal fluid, or CSF, and the chambers themselves are lined by the glial cell called an ependymal cell. Ependymal cells have cilia, so they're able to beat back and forth and move the cerebrospinal fluid around to bring nutrients and ions to the surrounding neuronal tissue. You have 4 ventricles. They are continuous with the central canal of the spinal cord, and the first and second ventricle are actually paired C-shaped lateral ventricles located separately in the left and right cerebral hemispheres. The third ventricle is located around the diencephalon, and the fourth ventricle is found at the base of the cerebellum or behind the pons. Now we're going to take a closer look at the adult brain regions. Let's start with the cerebral hemispheres. There's 3 parts to the cerebral hemispheres. We've talked about most of them. We have the outer or superficial gray matter called the cerebral cortex, the deep white matter, and then the deep gray matter called the basal nuclei. We also see that the cerebral hemispheres are kind of wrinkly looking. It contains ridges called gyri and grooves or valleys called sulci. Separating the left and right cerebral hemispheres are the fissure called the longitudinal fissure. The transverse cerebral fissure separates the cerebrum from the cerebellum. So here we can see the transverse fissure, which is a deep groove separating the cerebrum from the cerebellum. In this next image, we can see the longitudinal fissure, which again is a deep groove separating the left from the right cerebral hemisphere. Next, we want to talk about the 5 major lobes. There is the frontal, parietal, temporal, occipital, and insular lobes of the brain. The insular lobe is deep to the temporal lobe. We can see the temporal lobe in purple, and there's 2 temporal lobes, one on the other side as well. There's one occipital lobe in the light blue in the back of the head or brain, and then there's 2 parietal lobes. We're seeing the left parietal lobe. There'd be another one on the other side separated by the longitudinal fissure, and then the frontal lobe is there in pink. The frontal lobe and parietal lobes are separated by a slightly deeper groove. It's not a fissure, but it's slightly deeper than other sulci. And this groove is called the central sulcus. In front of and behind the central sulcus are important gyri. The precentral gyrus is in front of the central sulcus, and the postcentral gyrus is behind the central sulcus. So let's point out those again. Here is the central sulcus, a slightly deeper groove than the other sulci. It's separating the frontal lobe from the parietal lobe, and this bump in front of the central sulcus is the precentral gyrus, and that continues all the way down here. And on the other side, the bump behind the central sulcus is the postcentral gyrus. And they're going to have roles in the motor and sensory signaling or integration of the brain. Here we can see a better view of the gyri of the insula. We've had to move the temporal lobe down. Next, we're going to look more at the cerebral cortex. And if you've already forgotten what the cerebral cortex is, there's its definition: superficial gray matter. And it actually accounts for about 40% of the mass of the brain. So there's a lot of cortical neurons located in the cerebral cortex. And that's because it's really important for the integration of information that is involved in not only sensation, but also voluntary motor movements. This also includes communication, memory, and understanding. And each hemisphere is able to act contralaterally, or controls its opposite side of the body. What that means is that the left hemisphere controls the right side of the body, and the right hemisphere controls the left side of the body. But even though the hemispheres are acting contralaterally, they can communicate via the corpus callosum, that white matter band that connects the two hemispheres. So the hemispheres are not going to be equal in function, but neither one works alone. So no functional area acts alone. Conscious behavior is going to involve the entire cerebral cortex, or all of that superficial brain matter located on both the left and right hemispheres. Some functional areas of the cerebral cortex are things like motor areas, sensory areas, association areas. So these are the functions of the cerebral cortex. There's parts of the cerebral cortex that receive sensory information that make us aware of sensation consciously. And then there's areas of the cerebral cortex that are responsible for kind of getting an output signal together and controlling voluntary movement. Voluntary movement is movement of skeletal muscle or contraction of skeletal muscle to move our body. So that would be voluntary movement. So the effector for these motor areas is skeletal muscle. And then the association areas are very important. These association areas are pretty complicated if we were really to get into it, but their basic function is to integrate diverse information, to take this sensory signal, maybe from your ears and from your eyes, to take all this information and put it together into kind of one bundle that then can be sent out as a motor output to cause an action in response to whatever sensory signal or stimulus made its way to the brain. All right, before we continue on and look at the motor areas and the sensor areas, let's just do a quick review of kind of how does the central nervous system work. So we know central nervous systems are brain and spinal cord, peripheral nervous system, everything else. We know that we have things like peripheral nerves, we got receptors, we got all types of stuff outside of the central nervous system, and that we have sensory and motor signals. That sensory signals go to the central nervous system, and motor signals leave the central nervous system. So the way that these motor areas and sensory areas are processing information should make sense with that previous kind of information that we've garnered from the nervous system over kind of that basic review that we started with in chapter 11. So what's going on is the brain is receiving sensory signals from the peripheral nervous system. They're arriving at the brain. The brain is using that sensory information, maybe from multiple modalities, from sight, from hearing. We could have both auditory and visual stimuli. And we're putting the input, the sensory information, together and sending it from the sensory areas to the motor areas. The motor areas are able to take that multimodal association material and then decide what to do about that information, decide on a motor output, decide on a signal to send to the effectors. Effectors are just organs like glands or skeletal muscle or maybe even smooth muscle and cardiac muscle, but we're deciding what to send out to the effector glands, the organs that do stuff, and they will either contract or secrete, and that will be after they receive their motor output. So with that being said, with this basic understanding of how the brain works, very simply how the central nervous system works, let's look more at the cerebral cortex. We'll start with the motor areas, and there's 4 that we just want to acknowledge here, which are the primary somatic motor cortex, the premotor cortex, the Broca's area, and the frontal eye field. These are all motor areas. The primary or somatic motor cortex is important in moving your skeletal muscles. And if we didn't know that and we had to use context clues, something we should kind of key in on is that somatic. Somatic, soma means body. So this is going to be a motor cortex for something that moves your body. So, and that's exactly what the primary somatic motor cortex does. It's involved in conscious control of precise or skilled voluntary movements of our skeletal muscles. So it would send a motor output to skeletal muscles as its effector. The premotor cortex is going to kind of take information from the primary motor cortex and really integrate it. It's going to deal with things that require kind of sequencing or step-by-step actions of our motor complexes. And so the premotor cortex is a region that selects sequences based on kind of prior experience or memory or maybe skills or acquired reflexes and puts together these movements in a blueprint so we can accomplish more complex tasks. The cerebellum does something kind of similar, and so the premotor cortex works really closely with the cerebellum. But typing or playing a musical instrument, which requires kind of sequenced motor movement, would be handled by the premotor cortex, but it would get its kind of raw data from the primary motor cortex. Broca's area is a special motor speech area, so it's responsible for speech production. Think movement of your cheeks and your lips and your tongue in order to kind of shape sounds coming up past your glottis or past your vocal cords in order to make language. And it's usually present only in one hemisphere, usually the left hemisphere. Next, we're going to talk about the frontal eye field, and this is just going to control voluntary movement of the eyes. Again, it's controlling skeletal muscle. Around your eyes, you have 6 extrinsic skeletal muscles responsible for movement of the eyes. So here I've just pointed out those areas. We can see our motor areas are listed right under a banner. We have the primary motor cortex located in the precentral gyrus. In front of the precentral gyrus is another gyri where the premotor cortex would be located. Then we have a general location for the Broca's area and a general location for the frontal eye field, but they're generally located only in one hemisphere, and the left is the most common. Notice that our motor areas are located in front of the central sulcus, and most of our motor areas tend to be in the frontal lobe. Here is just a lateral view and cross-section of the brain, so you can see that these areas go deep to the cerebral cortex, but not quite into the cerebral white matter. We also want to acknowledge that we can do some mapping with the cerebral cortex, and it's called the motor homunculus, but basically it's the entire body represented spatially along the primary motor cortex. It's just the primary motor cortex. And what you're going to see is kind of like a funny cartoon. But it's talking about which cells, called pyramidal cells, of the cerebral cortex control what part of the body. And if that part of the body on this cartoon that's coming up is large, there's going to be more pyramidal cells devoted to motor movement or control of that body part. And the process of somatotopy, or this mapping of the pyramidal cells of the primary motor cortex behind the premotor cortex, creates this character, creates this cartoon. And while the process of creating it was called somatotopy, this kind of neuronal mapping, what we're about to look at is the motor homunculus. I threw in the sensory homunculus as well. Very similar, it's just looking at the postcentral gyrus, or the primary sensory cortex. And what we're seeing here is on the left, the motor map of the precentral gyrus. And we see here the sensory map of the postcentral gyrus. As long as you know one's motor, one is sensory, then you know what they're looking at. What we're saying with this image on the left is that the fingers have more neurons devoted to them, or their control, their motor control, than, say, your elbow, which is really small here. So it's a funny looking diagram, but depending on how big things are, that's how many neurons are devoted to either their motor movement or sensory sensation. So both your fingers, whether it's motor or sensory, have a lot of neurons devoted to their control. Whereas your elbow, not so much, not a whole lot for intra-abdominal sensory sensation. Has to be a lot of pressure or a lot of pain for that to be sent to our brain and for us to consciously be aware of it. But we can see that on the left, the entire body is represented spatially in the primary motor cortex of each hemisphere. And then on the right, we have like the relative amount of location of cortical tissue devoted to kind of proportional distorted body diagrams for sensory information. So you can also see up here in the middle where we're getting this information from, or which two gyri we're actually looking at. So for red or motor, we're looking at the precentral gyrus, also known as the primary motor cortex, and for the one on the right, the blue, we're looking at the postcentral gyrus, also known as the primary somatosensory cortex. So primary motor cortex, we know it's located in the precentral gyrus. We know that in the cerebral cortex, there's specific types of neuronal cells that we're calling pyramidal cells, and their axons are making up corticospinal tracts that travel down the spinal cord. They travel through that deep white matter into the spinal cord. And what the primary motor cortex is allowing is for control of motor movements, specifically voluntary motor movements, such as that of skeletal muscle. Precise motor control is represented by the motor homunculus, and this is the cartoon or the caricature we just talked about. What it's showing us is that relative amounts or certain parts of the cerebral cortex are dedicated to controlling different parts of the body, and that the areas where we have the structures appear larger mean that there's more of the cerebral cortex dedicated to the motor movement of that part than the parts that appear smaller. A review for the premotor cortex. We know it's in front of the precentral gyri and is located anterior to that. It controls repetitious, learned, acquired motor skills. So riding a bike, playing the piano, or an instrument in general, that coordination of sequential actions is performed by the premotor cortex, which gets its raw data from the primary motor cortex. Let's review Broca's area. Broca's area is usually located in one cerebral hemisphere. It can be located in the right, but it's usually the left. It is a motor speech area, so we're talking about movement of stuff in the mouth, movement or direction of muscles of the tongue, for example, a lot of those glossal muscles. And so it's going to be active as one prepares to produce speech. All right, before we move on, don't forget that those motor areas were all located in front of the central sulcus, that those areas were in the frontal lobe. Now we're going to move back and start looking at the temporal, the parietal, the occipital. That's where we're going to find a lot of these sensory areas, things like the primary somatosensory cortex, which is located in the postcentral gyrus. Then we have the somatosensory association cortices, visual and auditory areas. They have primary and association cortices. There's also going to be olfactory, gustatory, and vestibular cortices with their own primary and association cortices. So we have probably more of our brain, we could say, dedicated to sensing sensory stimuli than sending out motor outputs. I've highlighted in this image below this list a couple of different sensory areas. So taste is another word for gustation or gustatory. Smell is another term for olfaction or olfactory. Anything having to do with vestibular, or if you hear vestibulocochlear, think hearing or balance. And very conveniently, the cortices for hearing are located over here near the temporal lobe or in the insula, so kind of over near where you think your ears would be. Also for vision, we find those cortices located in the occipital lobe, so you could remember the phrase, someone has eyes in the back of their head. So eyes in the back of the head, so that's where our visual cortices are. Then we have things like taste, which are also going to be in the insula. Then the primary somatosensory cortex is going to be behind the central sulcus in the postcentral gyrus, and then right behind that is the somatosensory association cortex. So our general sensation areas are located kind of opposite the primary and premotor cortex in the frontal lobe. So let's review the primary somatosensory cortex. It had a homunculus. We know the primary somatosensory cortex is located in the postcentral gyrus, and it's going to be receiving information from the body. It's receiving information from really parts of the body that tell us where the body is in space. It allows for spatial discrimination, allows us to identify, like, are we raising our hand? Are we sitting down? Are we writing? Because we're receiving information from the body to the primary somatosensory cortex about your skin. Did someone pinch you? Skeletal muscle, are they relaxed? Are they contracting? Are they being pulled taut across a joint? Are the joints bending or are they inflamed? Are tendons torn or is everything normal? But this information is arriving at or going to the brain, and it arrives at the postcentral gyrus, which is the primary somatosensory cortex. There's also a somatosensory association cortex located behind the primary somatosensory cortex, and it's going to integrate that information or take the information from the primary somatosensory cortex and basically form comprehensive understanding of the stimulus. So think of the primary cortices for the brain getting the raw data, and then all of the association cortices, like kind of like a premotor cortex, they're going to put stuff together. They're going to create sequences, or they're going to combine multiple sensory stimuli together so we can make sense of what's going on. It's great if a tendon is pulled taut or is tight, or maybe it's inflamed, but that doesn't mean anything to the areas that are receiving raw data. That information has to be sent to our association cortices. Our association cortices are really the ones doing this integration. So here's our first association cortex, the somatosensory association cortex, and it's going to really just allow you to know that when you, for example, move your arm and put your hand in your pocket, that there's stuff in your pocket. That's not kind of the raw data, but not just that there's stuff in your pocket, but based on past experiences like memories or kind of acquired reflexes, skills, or just habit, you can probably feel what's in your pocket and then figure out what it is. You can figure out if something's a coin, or maybe it's your keys, or maybe there's an old piece of candy and it's melting and we need to get that out of there. But your sensory information has to be integrated in these association cortices, and they're going to be integrated in familiar sounding association cortices. So for the primary somatosensory cortex, it sends its information to be integrated by the somatosensory association cortex. For the primary olfactory cortex, it'll send its information to the association cortex for olfaction. So we'll have similar names. You'll know which ones are getting raw data, which ones are sending that information to be integrated in an association cortex. But this integrator is going to allow us to determine size, texture, and the relationship of parts from that raw data that we're receiving about our body. So next, let's look at our visual areas. There are primary visual cortices and a visual association area. So the primary visual cortex is going to take information like you saw a pretty flower, maybe there's a dog running around, or you see a bird in a tree. It's going to take that information, allow you to recognize what you're seeing, appreciate what you're seeing. Maybe the flower is pretty, but you only know it's pretty after you send the information to the visual association area. Before, you may have just registered there is something in front of me, the light is hitting the thing in front of me, that information is getting sent to my retina, and then your primary visual cortex would say, hey, this person is viewing something. But knowing that what you're seeing is a flower and that it's pretty is all because of the visual association area and that integration that allows us to interpret visual stimuli. Things about color, form, movement. Is the wind blowing? Is the flower blowing? Is there one flower? Two? How many petals does it have? What color is it? Do we like that color? So that's all about the association. Let's talk about a homeostatic imbalance related to vision, and that would be damage to either the primary visual cortex or the visual association area. It'll produce different things. Damage to the primary visual cortex actually results in what's called functional blindness. Even if information can get to your brain, that part of your brain that receives the raw data is damaged. So you're not able to actually process that visual stimuli. So it's almost as good as not getting the visual stimuli. So that would be functional blindness that results from damage to the primary visual cortex. Damage to the visual association area, that would result in individuals who can see. And what I mean by they can see is the light stimuli are able to hit the retina. The retina is able to convert the light stimuli into chemical signals. Those chemical signals travel from your retina via your optic nerve to your eye, and it reaches your primary visual cortex, because nothing's wrong in this scenario with the primary visual cortex. Something's wrong with the visual association area. So this individual can receive visual information to their brain, but they don't really know what they're looking at. They may have only, depending on the severity of the damage, an idea that something is shaded, it's darker or lighter than something else. But these individuals who have damage to their visual association areas are unable to comprehend what they are looking at. So let's look at where these vision areas are. Again, we want to think we have eyes in the back of our head, so that's where our brain is going to be processing visual information. So we have the primary visual cortex right here, kind of located at the point of the occipital lobe. And then the visual association area is located all around it. And it would actually go on the other side as well, kind of circulate around it. Next, let's look at our auditory areas. We have the primary auditory cortex, so that's how we're getting raw data. And we have the auditory association area, and that's how we're doing our integration. So the primary auditory cortex is located on the superior margin of the temporal lobe. So think temporal lobe near your temple or near your ear. So it makes sense that this is an area that is processing auditory information. And the primary auditory cortex is receiving raw data about pitch, rhythm, loudness, whereas the auditory association area is taking that raw data and seeing if you recognize it. Is it a song you've heard before? Or is someone murmuring? Are they mumbling? Are they screaming? Are they humming? Is that a storm off in the distance? Is that thunder, or is it just a breeze that's going through the trees? But it lets you actually perceive sound and see if kind of past memories or experiences are able to be applied to the sound so you can figure out what it is. Another area we just want to mention briefly is Wernicke's area. Wernicke's area is a sensory area, but it is located in the auditory cortex. It is also, like Broca's area, normally located in one cerebral hemisphere. So let's look for those auditory areas. So we found hearing. We see our primary auditory cortex and our association area, and they're located over here in this temporal lobe, specifically on that kind of superior portion of the temporal lobe. All right, let's talk about olfaction next. So olfaction, we'll look at the olfactory cortex, located on the medial aspect of the temporal lobe. So it's medial, we can't really see on the outside of the brain. It's located near the rhinencephalon. We sometimes call this the primitive rhinencephalon because it involves parts of the brain that are involved in olfaction, but also kind of linking this olfaction to memory. We don't really have the primitive rhinencephalon anymore. We have kind of structures that remain, like olfactory bulbs and olfactory tracts. But through the course of evolution, most of our rhinencephalon in the humans have taken over kind of new functions, things like emotion, memory, really kind of becoming a part of its own system. And that system would be the limbic system. You can call the limbic system your emotional brain. But it is our association cortice of the olfaction that's going to allow us to be aware, not only aware of odors, but to perceive them. But our primary olfactory cortex just lets us know that we're getting some raw data from our olfactory bulbs about the fact that we smell something. So primary olfactory cortex, we've actually removed the temporal lobe here so that we can see it because it was medial to that. It's located kind of in this curly-Q purple thing. That is a primary sensory cortex for olfaction. And there would be an association cortex, and that would let us know kind of what are we smelling, do we like the smell, do we hate it, have we smelled it before, that type of thing. Someone burning a candle, or did our dog just fart and we need to let them out. So next we're going to look at the gustatory cortex. So the gustatory cortex is actually in the insula. It's in that lobe that is deep to the temporal lobe. And the primary gustatory cortex is going to allow for the kind of, not perception of taste stimuli, not what something tastes like, but just the fact that taste stimuli have been received in the mouth and picked up by the taste buds and the gustatory hairs, and that's traveled to the brain. But then it's going to be the association cortex that's related to gustation that allows us to perceive something salty or sweet. Do we like the taste? Do we hate it? Did it taste like it smelled? Things like that. Then we have the visceral sensory area, and the primary cortex lets us know that something's going on in our GI tract or maybe urinary system. It could be a full bladder, an upset stomach. These visceral sensations, so visceral like organs, they would arrive at the central nervous system. But if we haven't experienced them before, we might not have a whole lot for our association cortex to do other than to tell our brain that it's painful or it's uncomfortable. So that would be an association kind of integration signal. But an upset stomach or full bladder, we may be able to use our association cortex to say, well, I just ate an entire pie, so my stomach's probably not really happy with me. Or I drank 5 bottles of water really fast, so that's why my bladder is full and now I have to go to the bathroom. But that would be information we use kind of other association areas in the body or in the brain for sensations in the body, as well as the association cortex related to visceral sensation. Next, we're going to look at the vestibular cortex. So think vestibulocochlear, the nerve that comes from the ear. But vestibular generally has to do with something about balance or equilibrium. So it's in the posterior part of the insula. It is adjacent to the parietal cortex, and it's just responsible for conscious awareness of balance, and it keeps our head kind of in position in space. So if you're driving in a car and you suddenly stop, that sensation of still moving forward is being processed by the vestibular cortex. All right, let's look at some of these areas. So we've already looked at hearing and vision. Taste, we talked about it's pointing to kind of medial or in the insula, medial to the temporal lobe. And then general sensation picked up by the primary somatosensory cortex and the somatosensory association cortices. And there are parts of that deal specifically with, for example, gustation and other things like that. Next, we're going to talk about multimodal association areas. And this is where things start to get a little complex. We still don't know exactly how multimodal association areas work, so that's kind of the best we could put together. So most of the cortices and association areas that we've covered thus far, they've really been linked to motor or somatosensory areas. We've been able to say this is a motor area or this is a sensory area. We can't say that for the multimodal association areas because they combine, they kind of put so much information together or make information that needs to go somewhere else that it's hard to dictate that they're one thing or the other. But they are generally located in areas where we can say most of the signals they're receiving are sensory or they're making motor outputs. But again, very generalized information we can give you about multimodal association areas. We do know that the multimodal association areas are complex. And this is due in part to the fact that they're receiving many sensory inputs from multiple senses, maybe 2 or 3 at a time, if not more. And they're sending out tons of motor outputs. And they're sending these to multiple areas, different types of effectors, glands and muscles at the same time, maybe smooth muscle and cardiac muscle at the same time. So multimodal association areas are complex in nature. So let's look at them in more detail. So they're receiving inputs from multiple sensory areas. They are sending outputs to multiple areas. And we want to include part of the frontal lobe called the premotor cortex as kind of something that's involved, we'll say, in the multimodal association areas. So we're involving a lot of sensory stimuli, and those need to be put in order, kind of put together, sequenced. So that's why the premotor cortex is involved. It's really great at that. So here is the general information flow in the brain if we include multimodal association cortices. So let's just take this step by step. So something happens in your body, and there's a sensory stimulus that can act on a sensory receptor. That sensory receptor picks up on that stimulus and sends a signal to the brain. So a sensory signal is going to go to the brain. It's going to arrive at the brain. That appropriate sensory signal will go to its appropriate sensory cortex. So if it's a sensory stimulus dealing with taste, it's not going to go to the cortex for hearing. It's going to go to its right sensory cortex. It'll go from the primary sensory cortex to its association cortex. So first the brain receives raw data, and then it integrates the information. From the sensory association cortex, next we'll send multiple sensory inputs to the multimodal association cortex. So let's take a break there. So say you hear a bird chirping. That sound wave hits your ear. It goes in your ear and travels via the vestibulocochlear nerve to the appropriate primary sensory cortex in your brain, then to the sensory association cortex. So you know that chirping, it's a bird. Then maybe you turn your head and you look at the tree where the sound's coming from and you see the bird. At the same time that you're seeing the bird, you're still hearing the bird. But now you see the bird, and that wave information travels as light and is able to hit your retina, and then your retina is able to process that information via your optic nerve and send that to your primary visual cortex. That raw data in your primary visual cortex then goes to the association visual cortex. So now you're hearing and seeing similar information. Your multimodal association cortex can take those two sensory stimuli that the brain has received and partially integrated and put them together so that we can come up with the idea, the conscious thought of the chirping I'm hearing is a bird and the bird is in the tree. So it's a very simple thought, but it's a complex pathway in order to form that conscious thought. After we use the multimodal association cortex, you may say that we're going to send information to the premotor cortex so that we can have kind of a sequence of events occurs. Maybe in order to see the bird better and to get more information, you kind of move so you can get closer, or you kind of move from maybe where you're seated to standing near a railing. Maybe you turn your head to get a better view or tilt your head. But this sequence of information is going to be handled by the premotor cortex, then sent to the motor cortex, which will send this raw data out to the effectors of your body, mainly somatic motor kind of areas, and those somatic motor areas would be skeletal muscle. So really what the multimodal association cortex is doing is allow us to give meaning to information we receive from multiple sensory stimuli. We may not do a whole lot of motor information other than store it as a memory, compare it to previous experience, or decide on an action to take. So every time you see, hear, smell, taste, we don't have to have a huge motor output, but something is occurring even if it's just the storing of that information. So all those decisions, that motor output or the idea that is made by the multimodal association cortex, has to be relayed to the premotor cortex and then communicated to the motor cortex in order for action to take place. All right, there's a couple of different multimodal association cortices, and we think that they're just pretty much what makes us who we are. They're areas where multiple sensations, thoughts, emotions, where they kind of become conscious. And one of the most talked about is the anterior association area, or your prefrontal cortex. So prefrontal cortex is one multimodal association area. There's also a posterior association area, kind of in the back, taking information from the other lobes. And there is your limbic association area, which is, again, limbic, think emotional brain. The prefrontal cortex is really complicated. We're looking mainly at the cerebral cortex of the front of the frontal lobe. So I'll make sure to point that out when we look at an image coming up. The prefrontal cortex is also going to talk to things like language areas, Broca's, Wernicke's areas. It uses general comment or interpretation areas, and it uses visceral association areas. So we're going to have kind of motor and sensory areas that are considered multimodal. So let's look at that anterior association area, or prefrontal cortex. So here is the central sulcus. So we know everything in front of that would be frontal lobe, also would be kind of the motor areas of the brain. And so the anterior association area, or prefrontal cortex, is just the front or most anterior portion of the frontal lobe. So that's what's there in red. The prefrontal cortex is involved in intelligence, learning, recall, so memory, and also personality. That big one there is personality. So the prefrontal cortex is involved not only in kind of logical learning, but personality. And it's necessary for judgment, reasoning, persistence, conscience, really things that we would use to describe someone. Are, do they make good judgments? Are they moral? Do they have good common sense? Are they stubborn? Or are they pretty chill? Things like that really are involved in describing a person's personality. And that's why personality is one of the functions of the prefrontal cortex. It's also closely linked to the limbic system, or the emotional part of the brain. And the prefrontal cortex doesn't fully develop until we're kind of in early adulthood. And that's one reason that preteens and teenagers are kind of abrupt in their actions. Things that they decide on may not make the most sense if we try to think of how they were linked together, because their limbic system and prefrontal cortex are not fully developed. A really interesting case where something happened to someone's prefrontal lobe, this kind of anterior association area, is the case of Phineas T. Gage. And you can actually see the injury over here on the right. So basically a steel rod went through this guy's prefrontal lobe. And so it gave us a lot of information about the prefrontal cortex really early on. Of course, we've developed that further. We know more information now. But early evidence that the prefrontal cortex played a role in personality really came from this case. So Phineas T. Gage was a foreman on a railroad, and he was doing some construction in the 19th century. And before this occurred, cases or interviews, now cases that were reviewed, reported that his personality was that he was reliable, organized, hardworking, things like that. And that changed when he was tamping in a iron rod into a hole drilled in rock, and he was using explosives and the charge unexpectedly exploded as he was packing in that explosive charge with this rod, and the rod went through his skull. And now you can see how it went through his skull. It definitely went through his prefrontal cortex. It actually was determined that it went through both the left and right prefrontal cortex. And after he recovered from this injury, which I know is surprising, but he was actually up and walking within a couple of hours, and in about 2 weeks, he was, for all intents and purposes, he was fully healed. But a lot of reports afterwards say that his personality changed. Or Gage was not Gage is one of the phrases seen in a lot of reports. He was no longer reliable. He was kind of impulsive. He wasn't as hardworking. He kind of became a drifter. And not much is known after he became an alcoholic and drifter, but the personality change kind of gave scientists a lot of information about what that part of the brain must do. So we know that the prefrontal cortex receives massive inputs from sensory association cortices, areas that are just general somatosensory, ones that are auditory, even visual. And this is really important as an area for higher order cognitive abilities, especially for personality, but also learning, memory, planning, attentional processing. And so a 1994 study did reveal with neuroimaging that Gage's skull, even though it healed, the placement of the injury indicated that he could have kind of exhibited emotional abnormalities in information processing, learning, and decision making. So definitely the case of Phineas T. Gage is interesting. His prefrontal lobe was damaged, his personality changed. Some of the first information we got about what that part of the brain does was from this case. All right, next we're going to talk about the posterior association area, which is a large area, not located just in the frontal lobe. There's parts of it in the temporal, parietal, and occipital lobes. And the posterior association area is going to let us know kind of where we are in space, but also let us recognize patterns and faces. So this is going to involve also understanding the written and spoken language. So this is a sensory area for speech. Broca's area is a motor area for speech. So language areas that we've talked about thus far are Wernicke's area and Broca's area. Wernicke's area is sensory, Broca's area is motor. There's also the lateral prefrontal cortex involved in word comprehension and analysis, and parts of the temporal lobe, which are involved in kind of auditory and visual aspects of language, like if someone's talking with their hands. There's also a general interpretation area. We think it's multimodal, but it's really ill-defined. There's parts of it in the temporal, parietal, and occipital lobes, usually also found in just one hemisphere, again, generally the left. And this would be able to integrate multiple incoming sensory signals into a single thought. So we think that there's a general area involved in conscious thought, but it may have to get information from other multimodal association areas. It's involved probably with the posterior association area, and it's going to be able to also process spatial relationships. All right, finally we've reached the limbic association area. Whenever you hear limbic system, think emotion. And it includes structures like the cingulus gyrus, the parahippocampal gyrus, and the hippocampus. The hippocampus is a good one to remember, is a part of the limbic system. The limbic system is our emotional brain. We're going to look at it in even more detail later. But it's providing emotional impact that makes a scene important to us. So if we spill hydrochloric acid on our legs, it's going to be the system that says, danger, this is acid, it could be really dangerous, that pain sensation we're feeling is not a good sign. So the hippocampus is also able to take that information from an experience like that and make memories. So that's another reason it's important to remember the hippocampus is going to allow us to make or establish memories. The limbic association area is located in the anterior portion of the temporal lobe. It's going to link emotion with many sensory inputs. One thing it can link with sensory inputs is smell. So if you have a lot of memories or emotions about certain smells that remind you of a past experience or have just been positive in your life whenever you smelled something, that's because of the link between your olfactory cortices and your limbic system. But it's also important in learning and memory. All right, so let's just take a minute, let that other information process, and let's do some review. Some of these terms are new, but the information isn't, such as the term lateralization. We've seen the term contralateralization, and contralateralization lets us know that it's the opposite sides of the body controlled by certain hemispheres. But the fact that hemispheres control the body is called lateralization, where each hemisphere has abilities not shared with its partner. We know the type of lateralization, again, for the brain is contralateral. There is a little bit of cerebral dominance, so the left hemisphere is better at kind of logical thought, math, language, and then the right hemisphere is better at what many people consider kind of the artistic side or artistic things, emotions, spatial skills, artistic skills. But they don't really work by themselves. Someone's not left or right-brained. Their personality may be such that it seems like they are, but the left and right hemispheres work together. So someone who's logical-brained or creative-brained, that's an oversimplification. Their cerebral hemispheres are working together. They just have parts of personality that may fire parts of the left hemisphere or right hemisphere more. So the two halves of the brain are going to be physically identical at birth, but different childhood experiences will utilize one hemisphere more than the other. But again, because lateralization, no hemisphere works alone, and we combine a lot of sensory stimuli and different modes of thought and conscious thought that are created to create our motor outputs. So sequential, verbal, analytic, logical, that's usually associated with the left hemisphere, and when neurons on that side fire, we're generally processing that type of information. Whereas holistic, visual, metaphoric, and intuitive, that information is generally processed on the right. But that information processed on one hemisphere must be communicated to the left in order for kind of efficient motor output to be created. So instead of saying that someone's left or right brain, you can just acknowledge their personality traits, but make sure you remember that the hemispheres work together to accomplish highly complicated sequences of sensory relay, integration, and motor output. Another interesting study is a study in lateralization, and we're going to look at split brain patients. Now, these individuals are interesting. They underwent what's called a corpus callosotomy, which is severing of the corpus callosum, which is white matter that connects the cerebral hemispheres. So essentially, their cerebral hemispheres were separated. So the severing of this structure led to really interesting behavioral phenomena. It really allowed researchers to see what was the point of each hemisphere, what one did over the other. And again, it's important that the hemispheres work together. So there were some things that these individuals were no longer able to do, or kind of took some workarounds. So we know language is primarily localized in the left hemisphere, so speech production. And the right hemisphere is mainly capable of just kind of basic language processing. But you can't use the right hemisphere neurons to produce speech, just kind of understanding it. It's more sensory. And what was observed was that if a patient was presented with an image to his left visual field, so going to the right brain, he would say he didn't see anything because the right brain is not really able to produce speech. So he wasn't able to verbalize what he saw because it was being processed by his right brain. But if he was able to feel around for certain objects, he could actually pick out the correct object, because while he couldn't verbalize the information, therefore his brain kind of panicked and said he didn't see anything, he did actually see something. And the right brain is able to process spatial and kind of texture information related to objects, and he could pick out the right object, even though he said he couldn't see the word of what he was shown, or he couldn't see the object of what he was shown in his left visual field. So different types of verbalization were required from these split brain patients if they were shown something in their left visual field only. But remember, a lot of times you wouldn't be in a situation where you only see something in your left visual field, or where you couldn't move your head to see it in your right visual field. So it was these controlled experiments that demonstrated what the left and right hemispheres did separately. There's another example where right-handed patients, after corpus callosum resectioning or splitting the brain, were able to draw with their left hands better than their right hands, because it was the right hemisphere that controls the left hand. So that's even if they were originally left-handed or originally right-handed, it didn't matter. It was going to be that they were able to draw better with their left hands than their right hands because of how information is processed in the brain. So just to review, let's look at the corpus callosum. Corpus callosum is cerebral white matter that connects the two cerebral hemispheres. So these green fiber tracks are just to show you where it's located, but it's cerebral white matter. So this would be just severed in a patient that was a split brain patient. Just more images so we can actually see a better picture here of the corpus callosum. Also, here are images of the basal nuclei. Over here we can see the basal nuclei, the dark or deep gray matter, and it's embedded in the deep white matter. Here they're outlined in different colors to show you the different types. Now, basal nuclei are masses of gray matter found deep within the cortical white matter. They do play a role in control of regulating attention and cognition. They're around the reticular activating system, or the RAS. And the degeneration of these basal nuclei we know causes Parkinson's disease, but we're not sure of all the links and how exactly its symptoms are produced. Better pictures of the basal nuclei. They would be surrounding the diencephalon, and we can see here part of the striatum that are involved in the basal nuclei. And then one of our last topics is looking at the diencephalon. The diencephalon is medial to the cerebral hemispheres. It's the central core of the forebrain, and it has three structures that make it up: the thalamus, hypothalamus, and epithalamus. Make sure you know that the thalamus is the relay station for information. It's kind of like the train station for information. Any information that goes up the spinal cord and is trying to get to its appropriate part of the cerebral cortex has to pass through the thalamus first. So it's going to route information to the right part of the cerebral cortex. So all inputs ascending to the cerebral cortex must pass through the thalamus. It plays a role in mediating sensation, motor activities, cortical arousing, learning, and memory. So impulses must be sorted out, edited, and relayed as a group from the thalamus to the proper cerebral cortex via the thalamus. The hypothalamus is located below the thalamus, hence the prefix hypo. It is connected to the pituitary gland via a piece of tissue called the infundibulum, which is called a stalk of the hypothalamus. So again, infundibulum, a stalk of tissue that connects the pituitary gland to the hypothalamus. The hypothalamus itself should be associated with homeostasis, and it has many functions. Some of its functions include control of the autonomic nervous system, so it's the boss of the autonomic nervous system. The autonomic nervous system controls contraction of cardiac muscle and smooth muscle, as well as secretions from glands, so it can regulate blood pressure, breathing rate, force of the heartbeat, digestive tract motility, rate and depth of respiration, and a lot of other visceral activities associated with your GI tract and organs in your abdominal pelvic cavity. It also is going to be linked to your limbic system, so it's involved in your emotional response. So it's involved, for example, in the perception of pleasure, fear, and rage. It deals with thermoregulation or body temperature control. Food intake and thirst intake are controlled by the hypothalamus, so it's your body's food and thirst center. It also regulates sleep and wake cycles by working with the epithalamus, which secretes melatonin, which is a circadian rhythm hormone regulator. And it's also going to be able to control the endocrine system. So the next chapter, after chapter 14 that you'll cover in AMP2, is the endocrine system, and we'll definitely see how the hypothalamus is linked to the production of many anterior pituitary hormones and how they are going to control the release of other hormones. And the endocrine system is a hormone releasing system. But again, H in hypothalamic or hypothalamus and the H in homeostasis should help you remember that the hypothalamus is involved in controlling many parts of the body and to make sure they maintain a homeostatic dynamic range. And one of the last parts of the diencephalon is the epithalamus. The epithalamus is also known as the pineal gland. It usually involves some other structures, but its primary one is going to be the pineal gland, which secretes melatonin. The pineal gland regulates the sleep and wake cycle, and it's going to work with the hypothalamus. Again, that was why regulating circadian rhythm was another goal or role of the hypothalamus. And that is it for part A of chapter 12. So next we're going to do part B, so stay tuned for that particular PowerPoint.